Welcome to the PubMed Daily Review for May 7th, 2026. Today we scanned 9,266 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Cerebrovascular Disorders and Treatment. This trend investigates mechanisms, treatments, and outcome prediction for diverse cerebrovascular disorders, from acute stroke to chronic atherosclerosis. This cluster covers 2,016 papers. Representative work includes theoretical cost-effectiveness of PCSK9 inhibitors in stroke due to intracranial atherosclerosis. Platelets cause microvascular occlusion and delayed neurological deficits after subarachnoid hemorrhage in mice, and prediction of malignant brain edema based on contrast agent exudation after mechanical thrombectomy for anterior circulation occlusion. Trend 2 of 5, Microbial Genomics and Engineering. This trend integrates microbial genomics and engineering to understand, track, and manipulate microorganisms and their biological systems. This cluster covers 2,549 papers. Representative work includes generation of cloned sheep lacking galactose alpha-1, 3, galactose, and N-glycolaminic acid antigens. Dynamic architecture of mycobacterial outer membranes revealed by all-atom simulations, and fully computational design of PM-relaxed Staphylococcus aureus Cas9 with expanded targeting capability using uni-design. Trend 3 of 5, Computational Cancer Therapeutics. This trend explores computational approaches to design, optimize, and assess cancer therapies, from drug development to treatment planning and patient outcomes. This cluster covers 968 papers. Representative work includes FilmQ-Dose, open-source software for radiochromic film dosimetry. HER2-driven mammary tumorigenesis enhances bioenergetics despite reductions in mitochondrial content, and synergistic effects of DNA-PKCS inhibition and radiotherapy in esophageal squamous cell carcinoma. Trend 4 of 5, Clinical Interventions and Outcomes. Research evaluates clinical interventions, diagnostic tools, and various factors affecting patient outcomes. This cluster covers 1,899 papers. Representative work includes routine long head of the biceps release improves pain and functional outcomes after arthroscopic rotator cuff repair of degenerative tears. A retrospective comparative study, transformed but not cured. The ethics of describing gene editing therapy for sickle cell disease, and Guardian presence in research conducted on children with blunt abdominal trauma in the emergency department. Trend 5 of 5. Human behavior and societal impact. This trend investigates the neural, cognitive, and social mechanisms driving human behavior and its profound societal consequences. This cluster covers 1,834 papers. Representative work includes conditional cash transfer and mortality among interpersonal violence victims, a cohort study, rethinking mandated drug treatment, why expanding freedom requires structural drug policy reform, and musical performance and biomedical human enhancement, ethnographic perspectives on bioethical questions. Up next, our first paper out of 5, a synthetic opsin restores vision in patients with severe retinal degeneration by Summerindra K., Mohanty, and Vineet B. Mahajan from Nanoscope Therapeutics Inc., 2777, North Emmons Freeway, Dallas, Texas, 75207, USA. Let's break down its key contributions. Imagine losing your sight due to inherited retinal degeneration, a leading cause of blindness. Current gene therapies often fail in advanced stages because too many cells are lost. Optogenetics, using light-sensitive proteins, offers a glimmer of hope, but existing technologies are too slow and imprecise. This paper explores a groundbreaking solution to overcome these challenges. This was an investigator-initiated, open-label study involving 4 patients with severe retinitis pigmentosa. They received a single intravitreal injection of a specially engineered AV2 gene therapy, delivering a novel synthetic opsin directly to their retinal cells. The study tracked their progress over 52 weeks, looking for improvements in their vision and daily function. The results were truly remarkable. All 4 patients experienced improvements in their vision, along with enhanced shape discrimination and mobility. Importantly, non-invasive imaging confirmed that the gene therapy successfully expressed the new opsin in their retinas. And perhaps most critically, there were no significant safety issues reported throughout the 52-week follow-up period. This study represents a major leap forward, demonstrating the first gene monotherapy capable of restoring vision in blind patients using optogenetics, and crucially, it's mutation-independent. The engineered synthopsin successfully addresses many limitations of prior opsin technologies. While these early results are incredibly promising, it's important to remember this was a small, open-label study, and larger, controlled trials are now essential to confirm these findings and bring this therapy closer to widespread clinical use. Up next, our second paper out of 5. Enable CRT cell immunotherapy in glioblastoma by modifying its microenvironment via oncolytic adenovirus, encoding bispecific T-cell engager by Moon-Young Choi and Olin D. Leong from Legoretta Cancer Center, Brown University, Providence, Rhode Island, 02903, USA. Let's break down its key contributions. Glioblastoma, a highly aggressive brain cancer, remains incredibly challenging to treat. While CAR T-cell therapies offer a glimmer of hope, their effectiveness is often thwarted by the tumor's ability to suppress the immune system and the difficulty for immune cells to cross the blood-brain barrier. This paper investigates a novel approach to overcome these critical hurdles and unleash the full potential of CAR T-cells in glioblastoma. Researchers designed a multimodal strategy, starting with in vitro studies using glioblastoma cell lines and advanced 3D models, including a blood-brain barrier spheroid. They then moved to an in vivo glioblastoma xenograft mouse model. The core of their approach involved an oncolytic adenovirus engineered to produce a bispecific T-cell engager, combined with CAR T-cell therapy. They meticulously analyzed T-cell activity, tumor cell killing, and tumor growth. The results were compelling. The oncolytic adenovirus, encoding the bispecific T-cell engager, or OV/T, significantly boosted T-cell recruitment to glioblastoma cells. This combination also markedly enhanced CRT cell infiltration and tumor-killing ability, even across a blood-brain barrier model. Most importantly, in a mouse model, the combined OV/T and CAR T-cell therapy led to a significant reduction in tumor size, outperforming either therapy alone. This suggests a powerful synergistic effect. This research presents a groundbreaking multimodal immunotherapy that effectively tackles the major obstacles in treating glioblastoma. By combining an oncolytic virus with a T-cell engager and CAR T-cells, the team has paved the way for a more potent attack on these aggressive brain tumors. While these are preclinical findings, they offer significant hope and a clear path for future clinical development, potentially transforming treatment for glioblastoma and other solid cancers. Up next, our third paper out of 5. In vivo CRISPR-mediated activation of cardiogenic genes to reprogram cardiac fibroblasts by Sachandra Dutta and Xiaoyang Liang from Department of Pathology and Laboratory Medicine, College of Medicine, University of Cincinnati, Ohio, United States. Let's break down its key contributions. Myocardial infarction, or heart attack, often leads to irreversible damage, where healthy heart muscle is replaced by scar tissue. This scar tissue, primarily formed by cardiac fibroblasts, can lead to heart failure. Imagine if we could reprogram these scar-forming cells into new, functional heart cells right inside the body. This study explores a groundbreaking approach to do just that, aiming to regenerate damaged heart tissue without invasive cell transplantation. To investigate this, researchers used a sophisticated mouse model where cardiac fibroblasts expressed a DCAS9 protein. After inducing a heart attack, they delivered adeno-associated viruses, or AAVs, carrying specific guide RNAs. These guide RNAs, part of a CRISPR activation system, were designed to switch on a set of key cardiogenic genes, essentially telling the fibroblasts to become heart cells. The goal was to see if this genetic reboot could transform scar tissue into functional cardiovascular cells. The results were exciting. In lab tests, the CRISPR system successfully turned on the target cardiogenic genes in fibroblasts. More importantly, when applied in mice after a heart attack, the activated fibroblasts began to transform into cells resembling cardiomyocytes and vascular smooth muscle cells. While overall heart function didn't reach statistical significance, the treatment did improve key hemodynamic parameters, effectively preserving the heart's performance compared to untreated controls. This study offers a groundbreaking proof of concept. We can potentially reprogram scar-forming cells into new heart cells directly within the body using CRISPR technology. This non-invasive approach could revolutionize how we treat heart failure. bypassing the complexities and risks of cell transplantation. While more work is needed to boost the efficiency and achieve full functional recovery, this research opens a powerful new avenue for cardiac regeneration. Up next, our fourth paper out of 5, an ADR2 mimic base editor for efficient C-to-U RNA editing in vivo by Chenhui Hao and Pei Hao from Aki Laboratory of Synthetic Biology, State Key Laboratory of Plant Trait Design, CAS Center for Excellence in Molecular Plant Sciences, Chinese Academy of Sciences, Shanghai, 200032, China. Let's break down its key contributions. Genetic diseases often stem from single-point mutations, and RNA editing offers a powerful way to correct these errors without altering the genome. However, existing RNA editing tools have significant drawbacks, including unintended DNA changes and potential immune responses. This research set out to develop a new, safer, and more effective RNA editing technology. The researchers designed a new tool called AMBER, an ADR2 mimic base editor for RNA. This innovative system was engineered to emulate the natural process of adenosine deaminase acting on RNA, or ADR, to achieve precise C-to-U RNA editing. They rigorously tested AMBER's performance across various cell lines and further evaluated its efficacy in a living mouse model. AMBER delivered impressive results, showing robust editing efficiency across a wide range of targets in various cell lines. Crucially, it also demonstrated strong efficacy when tested in a mouse model. A significant achievement was its ability to overcome the common challenges of concurrent DNA mutations and the immunogenicity associated with previous RNA editing tools. This study introduces AMBER as a highly promising therapeutic tool for repairing T-to-C mutations, offering a significant leap forward in RNA editing technology. By avoiding DNA mutations and reducing immunogenicity, AMBER addresses critical safety concerns. This breakthrough opens new avenues for treating a wide array of genetic diseases, though further research will be essential to translate these exciting findings into clinical applications. Up next, our fifth paper out of 5, Enhanced Multi-Cancer Screening Assay through Whole-Genome Methylation Sequencing-Based Multimodal Cell-Free DNA Analysis by Songmin Zhang and Taeyu Kim from IMBDX Incorporated, Seoul, Republic of Korea. Let's break down its key contributions. Cancer remains a leading cause of death globally, and catching it early is key to better outcomes. However, current screening methods often fall short, especially for detecting multiple cancers at their earliest, most treatable stages. This paper introduces an exciting new approach to tackle this challenge. Researchers developed a new assay called CancerFind, which uses whole-genome methylation sequencing to analyze cell-free DNA in blood. This innovative approach combines 4 distinct cfDNA characteristics, including methylation patterns and fragment sizes, using an advanced machine learning model. They tested this assay on over 1,400 samples, including patients with 8 different cancer types and healthy controls, to see how well it could detect cancer and identify its origin. The results were impressive. The assay achieved an overall sensitivity of 93.2% and a specificity of 95% for detecting cancer. Crucially, it showed excellent performance even for early-stage cancers, with 92.3% sensitivity for stage I and 92.2% for stage II. Furthermore, the model could accurately identify the tissue of origin in 85.7% of cases within the top 2 predictions. This study represents a significant leap forward in multi-cancer early detection. By combining advanced sequencing with machine learning, this assay offers a non-invasive, highly accurate method to detect cancer, even at its earliest stages. While these findings are promising, prospective studies are essential to validate its performance in real-world clinical settings. Ultimately, this technology has the potential to transform cancer screening and improve patient survival. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.